So now moving on to our next topic, where we will be talking about multiomics. It's an approach, it's an evolving science which investigates the genetics and the gene-environment interaction. And it combines genomics, epigenetics, microbiome, proteomics, and metabolomics. It combines the data from all these various aspects. And the idea is, again, to find answers for various unanswered medical conditions, and among which one of them is autism. And we have Dr. Chandrashekhar today with us to talk about this topic. He's the founder of Providence Microbiome Research Center and Resplice Autism Research Foundation. He's a principal investigator for the ongoing clinical trial on fecal microbiota transfer therapy for autism. So a very warm welcome, sir, and over to you.
Good morning, everyone. I'm Dr. Chandrashekhar. So I would like to discuss about role of multiomics in autism care and autism prevention. So everybody might be wondering what is multiomics. Multiomics is like it's a new advanced science. It's I can't call it as new advanced science, but this science is there from almost like last 30-40 years, but new technology that is that will enable us to diagnose the things at the molecular level. So this is an evolving science that can map all the biochemical pathways in the human body. So multiomics actually this is a combination of testing of the genetics and microbiome and epigenetics. Epigenetics is like functional genetics. And its descendants like proteomics and metabolomics, what are the entire chemical biochemical pathways can be made available. It's like this will work at the level of molecular level, and if you make a healthy profile of biochemical pathways, if you identify the healthy biochemical pathways, if you come across any patient which is like whether it is autism or Alzheimer's or any disease, cancer, anything, you map their biochemical profile and you compare these two profiles. You will understand what biochemical reactions are going wrong and what is which are the possibilities for that, and what is the precise medicine that can be prescribed. So this is the advantage of this one. This is right now it is very expensive, but this is the future medicine in the future. Like we will be heading for this precision medicine in the future. So let us understand this particular approach in detail. So you can see the picture. In this picture, this is an AI generated picture. I tried to generate with the AI. So this is a human body simulator. Human body is a cylindrical shell-shaped structure which has well-coordinated complex orchestra of biochemical pathways, so which we call it as physiology. A lot of physiological processes are there in the human body. All these physiological processes are organized in different organs of the body. So what is the basis of these physiological processes? So when you go deeper into the understanding and analysis of these complex orchestra of biochemical reactions, you will find in the deeper when you go to the ground level, all these biochemical reactions are being run by human DNA, and there is a part of essential microbiome in the human body. It is like the entire human body is covered by skin and mucous membranes. So the skin and mucous membranes have a covering of microbial layer, which we call it as microbiome. Be it as a skin microbiome, be it as upper respiratory tract microbiome, lower respiratory tract microbiome, and vaginal microbiome, uterine microbiome. Many microbiomes are there. All this microbiome has a part of microbiome is essential microbiome. The rest is commensals, but some part of the microbiome is integral part of the human body. The microbial genome, this essential microbial genome and human genome, they work in unison and run all the biochemical pathways in the human body. Whenever these biochemical pathways, whenever this association faces some disruption, then the biochemical pathways tend to deviate and the disease process starts. Whenever it progresses, it manifests as a major disease. So how it happens, let us see. There are three major components like human genome, the essential microbial genome. When these two association is met with positive environmental factors, then there will be healthy epigenetics. Like the gene to this human genome and microbial genome interaction and its functioning, how it is being working. Functioning of these two genomes is called epigenetics. Positive environmental factors, there is a positive epigenetics. Then, when the positive epigenetics is there, then the baby development and the functioning of the body will be healthy.
Dr. Chandrasekhar, sorry to interrupt. There is some echo on your end. Can you please check?
Okay.
So your voice is not coming out clearly.
So my voice is not clear.
There is some echo.
I'll check it and give you.
Sure. So while he joins back, if you have questions, please leave your questions in the chat.
Can you hear me?
Yes, sir. Yeah, please go ahead.
Now there is no echo?
No.
Yeah, okay. So we can see when this particular genome association between the human genome and this essential microbial genome encounters a negative environmental factors, that is, be it pesticides, be it antibiotics, and be it any other industrial toxins or anything, then the epigenetics gets altered. When the epigenetics gets altered, there will be pathological process going on. The biochemical pathways tend to deviate and they turn into disease process. When this happens during pregnancy, there will be development levels. So in this particular approach, you can see, so there are like human genome and essential microbial genome and environmental factors. In these three factors, there are two major modifiable factors, that is essential gut microbial genome and environmental factors. The human genome, which is not modifiable right now, but in the future, there are treatments like gene therapies and CRISPR are coming, but it may be a very long future. At this point of time, we have two modifiable factors, that is essential microbial genome and environmental factors. So these are the list of investigations in this particular modality. Like to test the genetics, we need to do exome sequencing and genome sequencing. Like exome is like what is the coding part of the entire DNA. Genome like the entire part of the DNA. And gut microbiome analysis. We can do this is called as metagenomic study. And toxic burden study, you can recently we started developing this test. We do not have an objective test to see what kind of toxic compounds we have inside our body. Now we are developing this test and it is awaiting validation. And the epigenetics part, we can do whole transcriptomic analysis and small RNA analysis, which is like regulatory genes kind of thing, and epigenetic mechanisms. Proteomics and metabolomics is like a manifestation of all these protein synthesis pathways. So, like we told earlier, there are two major modifying factors like essential gut microbiome genome and environmental factors. These are the two treatment targets. So to any diseases there which you are unable to answer, you go for a detox protocols and microbiome modulations. Microbiome modulation is like you can go for a dietary approach, you can go for prebiotics and probiotics, and when all these treatments fails and you don't want to go aggressive, you can go for fecal microbiota transplant, microbiota transfer therapy. So let us see how this is applicable in autism. So you can, if you understand autism in a molecular level, so if you take a autistic individuals autopsy studies, like if you take the brain studies or the postmortem brain tissue studies, you will come to know how many genes are going abnormal epigenetic mechanisms. So in the last conference, I discussed in detail about this one. There are around 584 genes they overfunction, and 550 genes they are underactive, and more than 800 genes they produce abnormal proteins or protein isoforms with mechanism called differential alternative splicing. This is only happening in the only brain itself. The entire body has around 5,000 genes are going abnormal functioning in the autistic child. So all this, most of the in these 5,000 genes also, many are the major problems are in the immune system and the neurotransmitter balance. These epigenetic errors during embryonic development and their physiology makes it a perfect case for the multiomics approach. So these are some of the abnormal epigenetic mechanisms during brain development. There are like upregulated gene expressions and downregulated gene expression, differential alternating splicing genes. You can see and you can test also, like now DNA methylation studies are available. All these kind of things can be seen. And so if you see postmortem brain tissue studies, so you will see these are some of the mechanisms like neuronal migration, neuronal differentiation, accelerated neurogenesis, microglial inactivation, and accelerated neurogenesis, and uh, functional circuitry formation. These are the important mechanisms that are affected. So when you go deeper into this one, like you will see that epigenetic errors are causing this problem. Because of this, there are microscopic structural abnormalities that are developed in the autistic brain. So this is happening in the embryonic stage itself, and we always tend to see the regression part and we think that the autism occurs after birth. It is not so. So regression is like an exacerbation of the neurotransmitter. So this happens when the child with autism uh, inherits a susceptible microbiome, and whenever they encounter with antibiotics in early phase and some vaccines also, we have seen they shatter down the existing susceptible microbiome that exacerbates neurotransmitter imbalance. So then regression happens. So this is like a so you can see this one. In this slide, we can see here this is the neural migration. So you can see the neuronal precursor cells migrate on this radial axis and form this cortical plate. So this is six-layer cortical plate. So there are like multiple. So you can see in this particular slide you can see these are some of the signaling proteins that are overexpressed. Because of that, there is over migration. So there is some sort of delayed migration also. So even the and this is called neuronal migration defects. And whenever you see other things like neuronal precursor cells, it has to mature into the mature neurons. So this is called differentiation. This process is also determined by some of the signaling pathways. Those are affected in autism. After the neuron develops into fully developed neuron, it tends to form connections with multiple connections with adjacent neurons. Only functional neurons, functional connections, like they will be further stronger, and any dysfunctional or missense synapses that will be uh, pruned, like they will be cut with the microglial activation. This mechanism is inactive in autism. So that's why they have excess number of synapses, which are missense. That's why they have abnormal circuitry formation. Because of that, they cannot process information properly. So this is like, if you see structural, what are the areas have these problems? Like these are the areas, like you can see, uh, these are the areas that are underdeveloped in the brain. And this is like, you can see it in postmortem brain tissue studies. These studies happened in the 1980s itself. So this is like we can see because of these microscopic abnormalities, children start developing this subependymal nodular dysplasia and the abnormal migration defect. Because of the abnormal migration defects, there is some sort of heterotopias, abnormal brain tissues. These ectopic brain tissues are generally trigger epilepsy. So, apart from this structural abnormalities, there is like physiological abnormalities also happen in because of the epigenetic errors. Four of them are important. Like one is like neurotransmitter imbalance. Second is like there is an abnormal immune response to the food antigens. Because of that, they develop a gut inflammation. Because of that, they have severe gut symptoms. Like it is similar to celiac disease. In the celiac disease, there is a gluten sensitivity there. Because of the gluten, they develop gut inflammation. And it is most of the children, if you do a food intolerance or food allergy test for the autistic children, you will find multiple food intolerances. These food antigens are not supposed to be triggering immune response in the human beings, but because of the abnormal gut microbiome, this gut microbiome, whenever this come across food antigens, that will report your immune cells that there is some sort of foreign antigen is. So if you have a good microbiome, that this process will not be there, then this food antigens will not be reported as a foreign body, and there is no gut inflammation. So having good gut microbiome is very much important to avoid this gut inflammation. And there is a subclinical neuroinflammation also. And because of this 5,000 abnormal gene expressions are there, because of that, there is a lot of functional deficiencies also there. That's why some supplements work for somebody, some supplements will not work for somebody, but supplements sometimes are needed. So how to approach for the autism care according to multiomics approach? If the autism is because of the genetics, we don't have much options for the genetics. So only gene therapies and CRISPR may be the future. And in the gene environment interaction part of the thing, so we have seen in the previous slides that genetics, essential microbiome, and environmental factors. For environmental factors, we can go for the detox protocols. In this detox protocols, also you can detox many of the things, but plastics, microplastic, no organism in this world has any mechanism to throw out the plastic that enters their body. So that is
Sorry to interrupt, sir. Can you just try going a little away from the mic?
Okay. So can you hear me now?
Yes, there is still some kind of echo. Maybe if you just go a little bit away from the mic.
Okay. Right now it is fine?
Yes, please go ahead.
Yeah. So the second thing is microbiome modulation therapies. So environment factors and microbiome modulation therapies, these are the two things that can help in autism. Like you can go for diet modification, prebiotics, probiotics, and postbiotics also there, and the fecal microbiota transplant. This is the most effective method to do a microbiome modulation. And for the functional deficiencies, you can go for the supplementation. These two, like environmental factors correction and microbiome modulation therapies, that will help bring some correction in epigenetics, and the child will become comfortable after that. So the regenerative medicine is also there, which can trigger the underdeveloped areas that can get stimulated and further mature. We can actually right now stem cells may be banned, but stem cells do have that capability. But we don't know the exact mechanisms where we can utilize this regenerative medicine. So what we are thinking of, we are doing some research to identify what kind of signaling molecules are there in the stem cells, which can be extracted, and we can administer as active signaling elements to allow that brain to underdeveloped areas to mature. So this is microbiota transfer therapy or fecal microbiota transfer therapy that I was talking about. We are doing this clinical trial since last 2 years. So we have seen there are two major mechanisms in the autism that are affected. That is abnormal immune responses and abnormal neurotransmitter, there is a neurotransmitter imbalance. These are the two things that are the major factors that actually make children suffer. So what we do is like we run detox protocols. Along with that, we do a microbiota transfer therapy, then we correct the epigenetic mechanisms. So there are two things you can see, like you can check for the food intolerance test and the urinary neurotransmitter imbalance, urinary neurotransmitter test. And if you see there are food intolerances and neurotransmitter imbalance in the urine, then you can go for this treatment followed by you can repeat the test and we can check it out. So this is like we are trying to establish this modality, and we are doing some ICMR approved clinical trials on this one. Once we submit this data, this may be available across India in the future. So this is for the autism care kind of thing and autism prevention. In the autism prevention, so I can tell you like 90% of children with autism are supposed to be born normal. So so this is like this may be vague, but all these children were supposed to be born normal. So they unfortunately lost their life. Human womb is becoming unfavorable normal childbirth now. So how it is happening a little. So we have to focus on at community level. We have to focus on prevention of autism. If you see, and 90% of cases, it is like every child has Tourette's syndrome, and 10% of children are autistic. 10% of autistic children have genetic component. If you test for genetics, most of the times you will not find any genetic. All this, if you don't find a genetic cause, that child was supposed to be born normal, but because of the toxicants that we ingested, whatever the toxins we have inside our body, is responsible for this autism. So you can see this slide. So yeah. So there are two major important environments. So in 2007, there is a study called toxic burden study from Environmental Protection Agency and Racial Network in the US. They studied the umbilical cord toxicology. So what are the toxic compounds that are entering the baby in different areas? So they are seeing like around 232 toxic compounds, which are mostly pesticides, weedicides, industrial toxins, microplastics. This is one of the major environmental factors that is affecting embryonic development. Another factor is there, like mother's gut microbiome. Out of the entire mother's gut microbiome, there is essential gut microbiome, like that is whether the gut microbiome itself is transferring or its microbiome is transferring into the child's colon, but we don't know. But so there is some sort of 18 tags of essential gut microbiome, which is species specific, is supposed to be transferred into the fetal colon. So we had a traditional understanding that uterus is sterile, and it is not so. Now lots of studies are coming up. Uterus is not sterile, but it is highly dynamic environment. In that highly dynamic environment, there are some only essential microbiome can survive. That essential microbiome, which is species specific, that go and colonize in the fetal colon, and that determines entire gut flora development. And it has direct role in the human entire fetal development. So this is happening like whenever the mother has a lot of toxic compounds in the body, then this microbiome is unable to colonize, and then it is affecting the entire epigenetics during the embryonic development. So this can be actually stopped. So this can be prevented. So whenever you are going for a pregnancy, you can do a food intolerance test to test the microbiome. So we tend to do a metagenomic study, microbiome studies like 16S rRNA sequencing, which is like most of the times inconclusive and don't know. We will not get any conclusive answer from that. But food intolerance test is a quality indicator test, and most of the times we understood that if somebody has food intolerances, they have bad microbiome. So if you go for a food intolerance test before pregnancy, and toxic burden test that we have developed now. That these are the two important factors. If you go for pregnant, if you do a test, these two tests before pregnancy, and detox yourself and correct your microbiome and go for pregnancy, the child will be healthy. So this is the campaign we are going to do in the future. So research is happening in this lines. So we have developed a ideal proposal for the autism prevention. That is like premarital genome matching. So 10% of autistic cases have genetic backdrop. So we are asking people to do a genome matching. So there is something called extended carrier profiling kind of thing that will prevent you. Like we always worried about thalassemia as well, thalassemia, hemophilia, and so many color blindness. Many of the cases, all these cases can be prevented with a premarital genome matching. Before marriage, you test your genetics, you match your genetics with your spouse, and then go for marriage. Then you will prevent this all genetic cases. And pre-pregnancy planning after the marriage also, you have to go for a pre-pregnancy planning in this particular toxic world now we are living in. That is toxic burden test. You do a toxic burden test. How much toxins you have deposited in your body, and go for detox protocols, and you text for the food intolerance, and then look for any microbiome disruptions. These two things you can see, and you can correct these two things, and then go for pregnancy. During pregnancy, also you need to go for a lifestyle modification. You should avoid cosmetics usage and pesticide containing food and junk food, and all these things. These should be avoided, and you like, should be very cautious about antibiotics and all. Any drugs or anything, smoking, alcohol, all these things have to be stopped. If you do a lifestyle modification and take proper antioxidants during pregnancy, then you can prevent autism. So take home message from my presentation is like for kids with autism. So we have a ready-made answers from the multiomics approach. You can it has to be proven now. So what we are going to prove it in 1 or 2 minutes next now. So you can do a food intolerance test and urinary neurotransmitter test, and opt for the detox protocols and microbiome modulation therapies, and supplements for the functional deficiencies, and then go for regenerative medicine also. This is a simple take-home message for the kids with autism. For the parents who are going for second pregnancy, if they have autism in first child, if you want to go for the second pregnancy, then you can test your genetics. Parents should be tested for their genetics, followed by mother should be tested for the food intolerance and toxic burden test. Then detox yourself and go for do a microbiome modulation, and then go for pregnancy. Then you will have a high possibility of healthy pregnancy and high possibility of healthy child. This is the thing. For this is like and another is like for the community. For the community is like we have to do it we have to do it in a great scale. Like it has to be taken up by the government. It should be like something like folic acid in the antenatal program we give for NNK eradication. Like we have to give some medicines like vitamin C and N-acetylcysteine, astaxanthin, or some antioxidants, and along with some probiotics like BSL-3 and these kind of things. If you introduce in the pregnancy during the growth pregnancy, the child will be normal. So you can actually, so now the technology has improved a lot. So you can actually thalassemia, autism, hemophilia, color blindness, and many genetic disorders can be prevented, and the autism also. You can bring it back to 1 in 1 lakh. So this is a possibility, and so the research is going on these lines in our center. So we'll prove.
Sorry to interrupt.
Yeah, please tell me.
Yeah, sir. So we are out. The time is over.
Yeah, this is the last slide, right?
Yeah. Yes.
So yeah, you can note this. So this is like. Yeah. So these are the differences. This is the website. This is our website, resplicecdc.com. So in that we have given all the references for these studies. You can go through that. And say no to pesticides during pregnancy. Thank you very much. And thank you IAFM for the conference, and it's a great conference. So thank you very much. Thank you all.
Thank you so much, sir. It was definitely very not with lots of knowledge, and your take-home message. I think that is something that's very important for all women out there who are planning for a pregnancy or who already have a first child. On the spectrum, it becomes even more important to prepare the body for the second baby. We have a lot of questions, and I request you to please stay back and answer them. And we will take a lot more questions during the panel. And
Sure.
thank you so much.
Thank you. Thank you so much. Thank you.